And I'd like to invite Yael Cohen from the Hebrew University to the stage.
So hi everyone. As the beginning of the second part, we've heard a few speeches. I would like to tell you that I've just come from a 2-day workshop from all kinds of fields. I think even broader than here. Here we have all sciences. My height, yeah. We have all sciences, and we had people from psychology and philosophy and history, all discussing the same very core question in science: is what makes us bias in science? What type of things make us make very, very big mistakes or sometimes smaller mistakes? And some of these things are financial, like what type of research is considered important. Sometimes it's looking to find the answers we want to find. If we look for a particular answer, we're likely to find it. Or what type of research is considered cool these days is also a reason for why sometimes we're biased. But I think the most fundamental reasons for being biased is something that we often blind to is what our axioms are, what our fundamental, down beneath what our beliefs are, our assumptions are, and that's what we build our research on. And I don't have good answers from this workshop, but I can say that one of the biggest, or one thing that we did see in the workshop is that in order to avoid these biases, the best approach I can think of to do that is to see people that think in a different way than we do. And a different way is what type of questions they ask, and also what type of approaches or what type of methods they use. So I'd like to thank you not only for supporting my research, but also for getting us all together and hearing many, many different people from different fields. I think it helps every single one of us to hear what other people are doing. So in a non-biased manner, I would like to tell you why what I'm doing is interesting, and I hope I'll convince you as well. I'm coming from physics, and I look at geometry of materials, of shapes. When you look at a bridge, you can look at what it's made out of, if it's a wooden bridge or made out of stone. But you can also look at the shape of the bridge, and the shape of the bridge influences how strong the bridge is. And the same is for the shape of flowers. How much sun they're exposed to comes from the geometry, and the functionality of cells in our body comes from their shape. And not only do we want to understand the shape, but we'd like to be able to influence the shape, because the shape is what helps us, influences functionality. And this is something that we all do on almost an everyday basis. When you have pizza, right? Everyone has a piece of pizza in their hand. And often when we hold a piece of pizza, the cheese just flows down. And to avoid that, we want to use the geometry of the slice of pizza to have it to have the cheese stay on the pizza. And for that, we fold the pizza, right? We curve the piece of pizza, we have it, and then it's a much stronger piece of pizza. And to do that, slice of pizza, to do that, we'd like to do it on all scales. This is from bridges to cells. This is something we'd like to influence the geometry of things. And before we influence it, we want to understand it. And yes, a bridge is the final shape or the flower, but it's built layer by layer or stone by stone. And if we look at knitted fabrics, for example, that often get different shapes, look at a scarf, it's knitted row by row or layer by layer with a final shape in mind. And what I do in my research is I look at these blocks, I look at two molecules and ask how they interact one with another and how those interactions influence the final shape of the assembly. So they connect one by one by one, and I want to ask what the final geometry is going to be like. And this might sound quite straightforward. If we have one and another, then we'd know the answer. But in physics, we say more is different, and different meaning fundamentally different. One ant is not the same as a whole nest of ants, right? They behave differently if you look at one or you look at the whole crowd. And looking at this looking at the two molecules and trying to find the final geometry is what I've been working on, what I'll continue working on. So thank you.
Okay. Thank you. And the next one is Jonathan Jung from Hebrew University.
So first, I'd really like to say what a great honor it is to receive this Adams Scholarship. So thank you, Mr. Adams and the Adams Foundation. I'm Jonathan, and I study biology at the Hebrew University under the guidance of Nissim Ben-Benisti. And I always had an interest in animals and nature. However, I grew up with two biologists as parents, and I was convinced that I would go to a totally different direction in life. And that actually changed only in high school, where my biology teacher didn't teach us by the normal textbook, but she actually brought scientific papers, and she would change the lessons and make them actually interesting discussions about new discoveries that are just getting out there. So this kind of pulled me back into the biology field, and now I can finally say that I understand the conversations around the dinner table. So yeah. But I focus on stem cells in my research, and stem cells are actually very, very remarkable cells because they give rise to all of the other cells in the body. And they give rise to the heart, to the brain, to the eye, and because of that, they are actually a very strong tool to use in researching the development of human, in researching diseases which are developmental diseases. And also, these cells can actually be used as cell therapies to be treated to patients. But there is a challenge. When we grow these cells in the lab, they are actually grown under conditions which aren't the natural environment that they would feel in the body. And like any other living system, when they are under pressure, they will adapt. And sometimes these adaptations are actually happening through mutations, random mutations. And these mutations can sometimes be mutations that are also found in cancer. And this matters because if we have these cells with these cancer mutations, they will not behave like we expect them to. And this will change our scientific results that we get from them. And even more so, think about putting these cells into the patient. Think about the consequence of that. So you might say, okay, but these cells, these mutations rarely happen, and because of that, it's very highly unlikely. But actually, I will say no. These mutations are quite common. And my research specifically is trying to understand how often do these mutations occur and understand what do these mutations actually do to the cells. And also I am trying to find ways to reduce them in culture. This is something that is on the way. But what we found is that 20% of the stem cell samples that we analyzed from around the world actually have these cancer mutations. And that is a critical number because it's much more than what we expected. And because of that, we have to start addressing it immediately and start making better regulations to how we grow these cells, how we evaluate them, and how to see that they're actually okay to be used. So in a world where stem cell use is starting to be more and more common, I hope that my research will actually ensure that it is done in a safe and responsible manner. And I'm excited to continue on learning and growing and making a positive impact on this rapidly evolving field. And thank you, and thank you, thank you.
Okay. I'd like now to invite Tomer David Keidar from Tel Aviv University.
Hello, everyone. My name is Tomer Keidar. I'm a PhD student in physical chemistry at Tel Aviv University. From a young age, I was fascinated by the world around us and by unifying laws in it. One thing that we can find almost everywhere is randomness. For example, when you stand in a queue for a random amount of time, or when an epidemic spreads through populations due to random encounters between healthy and sick individuals. In finance, we cannot predict which stock will rise and which will crash. In chemistry, the time for two molecules to meet and react is random. In biology, it controls the time and location of mutations on DNA. And in physics, we see it in the random erratic movements of particles submerged in liquid. But the central goal of science is understanding how a system responds when we intervene. For example, how an object will move after a force is applied. So how do such random processes respond to changes in their environment or operations? In most cases, we lack a general answer. In my PhD, I'm trying to bridge this gap. Today, I want to tell you about a recent project I led. I studied how the average time for something to happen for the first time changes when there is some intervention that happens at some rate. At first, it seems that finding the response of a general process to a general change will require detailed analysis of the problem. However, I showed that when this change happens rarely, the response is rather simple. It turns out that all the details are irrelevant, except for three things: the average completion times and the fluctuations around it, and one average time which related to what we're actually doing. And this is all the information we need to know about this change. I think that this result is interesting on its own, but its main importance comes from the fact that it enables a new kind of experiment, measuring molecules' lifetime fluctuations from bulk measurements. So imagine a biological cell that's producing some molecules, where each one of them is then degraded through some process. If you have a test tube full of those cells, and we measure the molecule concentration, we learn only about their mean lifetime, their average lifetime. The only method available to get more information, for example, the fluctuations around this average, was to catch individual molecules and track the lifetime of each one of them individually. And if it sounds hard, well, this is because it is. But as I told you, the concentration depends on the average lifetime. And its response to a rare change is also dictated by the fluctuations around it. So if we can affect the molecule's lifetime by a rare change, we'll be able to measure the fluctuations in their lifetime by measuring the response of the concentration to that change. One simple possibility is to add a small amount of annihilator molecule that it will annihilate our molecule of interest upon contact. Using computer simulations, I showed that this enables the inference of the fluctuations of the molecule's lifetime by measuring concentration alone. A much simpler and cheaper experiment compared to the single molecule experiments used today. I believe this study illustrates something beautiful about science, phenomena that appear completely different, from molecules inside a cell to epidemics spreading through a population, can sometimes obey the same underlying principles. By uncovering this principle, we gain not only a deeper understanding of nature, but also new tools for measuring and predicting the world around us. I would like to thank the committee members, the Israel Academy of Sciences and Humanities, and Mr. Sylvan Adams for this prestigious and generous fellowship, and of course my parents and my amazing partner Shir for their support. Thank you very much.
Thank you, Tomer. Next is Yeela Persky from Ben-Gurion University.
Hey everyone, so my name is Yeela, and I want to start by saying what a great honor it is for me to be here. And it's especially meaningful to me to be here today and to be a researcher in the chemistry field, because by doing that, I'm keeping my family tradition. Both of my grandfathers from both sides are researchers in chemistry, one in the industry and one in the academia. And I am very proud to continue the way. And I hope I can raise my young daughter to do not the same, but to pursue knowledge and to do something meaningful and donate to this world. So thank you, my family. And I want to tell you a little bit today about what am I doing as a chemistry researcher. I'm focusing on the field of energy. Like Elad here presented in the beginning, fossil fuels have many problems, as I'm sure all of you know. For example, as we saw in the last few months, the geopolitical influences on the gas price, right? So we want to overcome this problem by moving to renewable energy. Okay, we have many reasons to move to renewable energies, but we have one main problem, which is it's unreliable. So in order to trust this source and to have electricity all over the year and all over the day, we need solution for storing energy. What do I mean by storing? I need to take excess of electricity that I have during peak time when I have a lot of sun, for example, at noon, and store them in some kind of box, something physical that I can actually hold and store for a very long time. So one of the ways to do it is store the electrical energy inside of chemical bond. When I want to store my energy, I take my excess of electricity and create new chemical bond, build molecules. And when I want this energy back during, for example, at night, I take my molecule and break the chemical bond and get my electricity. One of the molecules that is really great for doing that is hydrogen. It's a really small molecule. So therefore, I get really high energy density inside of just a little bit of hydrogen. In order to use this hydrogen, I need a very common source for hydrogen. I'm sure you, maybe not all of you know, but you all do use it in your daily life. It's water. Water is composed of two kinds of atoms. We have hydrogen and we have oxygen. So when I have electricity, I can take the water, break it, and separate the hydrogen and the oxygen. And when I want my energy back, I take the hydrogen, take the oxygen, combine them together. I'm actually breaking the hydrogen molecule and getting my energy back. This process that I'm doing with electricity have a very high demand of energy. So in order to reduce this demand, we need materials that will help us do this reaction more efficiently. This material called catalysts. In my research, I'm trying to find new materials that will be good for this reaction, will make it more efficient, and will be cheap. So I'm working on new material design and also on new synthetic way that will be more energy and time efficient. And thanks to the Adams family and all the committee, I will be able to keep doing this research. So thank you for your support and for believing me. And I hope that in the future, my research study will find something that will be efficient enough and cheap enough, and we can storage energy and move to renewable energy for a better world. Thank you very much.
Thank you, Yeela. This is Maya Schlesinger from Tel Aviv University. Maya, here.
Hi, everyone. I'm Maya. I love games. Ever since I was a kid, I would play them a lot after school for hours on end with my friends, but not just video games, also Dungeons and Dragons for those familiar. I would spend hours building characters, trying to make them as strong as possible while managing limited resources, and I enjoyed it a lot. Thank you. I spent so much of my free time playing games, and I always felt a little bit guilty, like maybe I should be doing something more productive with my time. What I didn't know then, but I do know now, is that games can actually be important. They're how we model economics. And I think 12-year-old Maya would be, sorry, there's a lot of people here. I think 12-year-old Maya would be extremely happy to know that my career is researching games. So what do I mean by that? Games are the language of economics. Well, in games, we try to maximize our points, our power, whatever resource we're trying to accumulate while managing the limited resources that we have. That is exactly what economic agents like organizations are doing. They're trying to maximize their profits while managing limited resources. So I want to give you an example of the type of game that I study in my research. Imagine you're trying to sell a house. You can't be there all the time to show it to potential buyers and to negotiate, so you hire someone to do that for you. You hire a realtor. You've now entered a two-player game between you, the seller, and the realtor. So what is your optimal strategy in this game? Do you pay them a fixed salary until the house is sold? Or do you pay them a commission once it is sold? You all know that paying them a fixed salary is probably not such a good strategy for the game, right? They would just happily keep on taking your money, never actually making the sale, because they are self-interested. They have no incentive to sell the house. So you pay them a commission. Good. That's our optimal strategy, right? But wait, how high should the commission be? If you only give them, let's say, 0.1%, they're not very well incentivized for the sale value to be high or even to sell at all. If you give them 50%, you're losing out 50% of your revenue. There's a lot of considerations that go into this game, and these are studied by a field of economics called contract design. This has been a rich body of work that's been developed for the past few decades, even culminating in a Nobel Prize in 2016 to two of its founders. But our world is constantly changing, and sometimes the answers that we've already had don't suffice. So imagine you're no longer selling a single house and hiring a single realtor. Imagine you're the CEO of Uber, and now you have to work with 10 million drivers worldwide. How do you incentivize them? If you pay them too little, you won't have any drivers, and you won't have any business either. If you pay them too much, you'll go bankrupt. Do you need to pay them individually, a different amount to each one, depending on local conditions like gas prices? Should the ride matter? There's so many complex considerations that go into this large-scale system, and we need a computational approach. We need algorithms that are able to actually come up with good contracts in large, complex systems. This is the work that is now enabled thanks to the generous contribution of the Adams Fellowship, and in particular, thanks to Mr. Adams. I am very happy to continue researching how can we actually computationally find good contracts in complex settings. Now, you might be thinking, I'm not the CEO of Uber. I don't have a house I want to sell. Why should I care about contract design? Now, it won't surprise you that I'm not the CEO of Uber. I also don't have a house. Well, I live in one, but I don't own one. And I still care. Why? Because I'm not designing these contracts. I'm not playing in the game, but I'm affected by it. Contracts are all around us. Someone is paid to maintain the roads that you're driving on and make sure they're safe. Someone is paid to groom the parks that you go on and make sure that they're pretty and green and you can get your sunlight and happiness. And someone is even paid to educate your kids. So we're not designing these contracts in day-to-day life, but I think we all hope that the people who are know what game they're playing. Thank you so much.
Thank you, Maya. Our next next fellow is Aviezer Silverman from the Hebrew University. Aviezer.
Hi, everyone. Don't worry, I think I'm the last. So, good afternoon. I would like to start by saying that when I was 16 years old, I volunteered in Magen David Adom, the National Emergency Services. I would go mostly after school for shifts where I would sit in the back of an ambulance and manage medical situations. I did the basics like, measuring blood pressure or monitoring oxygen levels, but I also experienced many extreme medical situations. Not many teenage boys could say that they helped deliver twins. Now, this early on experiences taught me how complex the human body is. It started a deep desire in me to help other people and eventually paved my way to go and study or want to study medicine. But on my way there, I studied biomedical science and I completely fell in love with it. I learned how sophisticated nature is. It taught me early on how much is still unknown but left for us to study. So then I thought about it, and it came to my attention that there is so much work behind the scenes and work we need to do in order to help other people. And so currently I'm doing my PhD in microbiology in the Hebrew University, and I'd like to share with you a little bit what it is that I research. But before that, let me ask you a question. When was the last time you used antibiotics? So I'm willing to bet that most of us in this room have used some sort of antibiotic in the last year or two. It saved countless lives, probably mine and yours too. But what if I told you that this incredible tool is slowly losing its power? That's scary. I'm 30 years old and have 3 very cute children. A few months ago, my youngest daughter, Yael, she had fever, and it turned out she was suffering from a urinary tract infection. So some of you may know that before starting treatment, the doctor needs to send a culture to check what bacteria is growing. He will also learn what antibiotics he should use, but also which one he shouldn't. So most people, normal people, will just, get the prescription and start the treatment. But as a microbiologist, I couldn't help myself. I wanted to see the results. And I was shocked to see a long list of standard antibiotics that simply wouldn't work against this specific infection. Fortunately, there were still other options, and she recovered just fine. But that moment was a wake-up call to all of us. The bad bacteria are learning to overcome our best medicines. This issue, which we call antimicrobial resistance, is a growing problem in clinics and hospitals all around the world. Well, luckily enough, nature didn't leave us empty-handed. It turns out that there's a specific type of viruses that they don't affect humans at all. Instead, they target and kill bacteria. We call these good viruses phages. So phages could be used to kill the bad bacteria. The truth is, this isn't anything new. We've been known for almost 100 years about these phages, and we're even used to treat some infections. So what happened? Well, a few years later, we discovered the penicillin, which worked like magic. But today, as antibiotics stop working, it's time to revisit these phages and possibly give them another chance. And in my research, that is what I'm doing. I'm curious in understanding how these good viruses work. And to try and explain to you what they do, I'd like us all to imagine a big enemy ship sailing the ocean. This is our bad bacteria. Now along comes a good small pirate ship, maybe like Jack Sparrow on the Black Pearl. So instead of firing cannons and sinking this ship right away, he deploys undercover crew members onto this ship with very specific instructions. Their instructions are to take the controls of the ship, use its own resources to create new pirate ships, and only then to sink this ship and release into the environment new pirates. This is exactly how phages work, more or less. They hijack the bacteria, they overtake the replication mechanisms to create new of themselves, and only afterwards do they kill the host, kill the bacteria, and release into the environment new phages who could now go and infect other bacteria. I would like to extend my deepest gratitude to the Adams Family Foundation and specifically to Mr. Sylvan Adams for this tremendous contribution. This scholarship would enable my continuous research in understanding how these good viruses work. I truly believe that one day, if not us, then maybe our children and grandchildren will use these phages, these good viruses, to tackle life-threatening bacteria. And this is why my research is so important. We are constantly learning how these microscopic pirates do what they do best, and that is to kill bacteria. So thank you very much.
Thank you, Aviezer. And before moving on, I would also like to recognize Avichai Marmor from Bar Ilan University, who was not able to be with us this evening. And now we will continue with the Ruth Arnon postdoctoral fellowships. There aren't quite enough words to describe Professor Arnon's personality and incredible contributions to science. I have been fortunate to work in the same building with Professor Arnon, we call her Ruthie, for over 40 years. And my admiration for her has been growing every year. For me and for the scientific community, in Israel and abroad, she is a real role model. Her success in scientific research and her academic leadership have inspired and paved the path for many scientists, especially Israeli women researchers. Her academic achievements have awarded her many prestigious prizes and awards in Israel and abroad. Her contribution to medicine has changed countless lives of patients and doctors alike, and many of you probably know about Copaxone, which really has saved many lives of people suffering from a severe neurodegenerative disease, severe muscle disease, and this was developed in Ruth Arnon's lab, and she is now also pioneering new types of vaccines against influenza virus, so really major impact. Her intelligence, leadership, and generosity have made her both respected and admired in academic circles worldwide. Moreover, Professor Ruth Arnon's own distinguished career demonstrates that women can excel as professors and scientific leaders without compromising their devotion to family life. And hopefully, we also have some fellows, some Adams fellows here who will be happy to follow Ruthie's path in combining family life with excellence in science. The Ruth Arnon Fellowship Program, which she so generously established, gives female Adams alumni the support and encouragement to pursue postdoctoral research abroad and advance their own contributions to Israeli Professor Ruth Arnon celebrated her 93rd birthday just a couple of weeks ago. Unfortunately, she was unable to join us this evening. Hopefully, she will be able to join us next year. So I'm delighted to share a few words about the two awardees of the Ruth Arnon Fellowships, last year's and this year's, who are not able to be with us today because both of them are already abroad pursuing their postdoctoral studies, one in France and one in the United States. So our Ruth Arnon Fellow for the 25-26 academic year is Dr. Einat Seidel-Posner, a physician-scientist whose work bridges clinical medicine and immunological research. She earned her MD and PhD degrees from the Hebrew University Faculty of Medicine and completed her residency in internal medicine at the Hadassah Ein Kerem Medical Center. Her research has advanced our understanding of strategies that viruses use to evade immune detection at the molecular level. She recently began a postdoctoral fellowship at the Institut Pasteur in Paris. I would say the kind of mecca of microbiology, where Pasteur worked. Everybody knows about him, of course. And she's focusing on autoimmunity, and her long-term goal is to return to Israel as a physician-scientist, combining patient care with translational research in the field of rheumatology. Our Ruth Arnon Fellow in the 26-27 academic year is Dr. Dana Binyamin, a microbiome researcher specializing in host microbiome interactions, with a focus on how the microbial communities influence human health and disease. We heard very recently, right, about ways to target the bad bacteria in our bodies, and there are many more reasons to study microbes and ways to manipulate and modify them. She earned her PhD degree at Bar Ilan University, Faculty of Medicine, essentially in Tzfat, where she studied how the gut microbiota influences aging and host physiology, including its effects on metabolic processes. In parallel, she investigated microbiome dynamics in pregnant women with inflammatory bowel disease, examining host microbiome interactions and potential implications for disease susceptibility in their children. She recently began a postdoctoral fellowship at Stanford University School of Medicine, where her research focuses on understanding the ways in which the microbiome influences host physiology and disease. Her long-term goal is to uncover microbiome-driven mechanisms that can be translated into targeted therapies, advancing disease prevention and improving health through precision microbiome interventions. We wish our Ruth Arnon Fellows continued success in their research and look forward to welcoming them back in Israel as exemplary scientists and future leaders of our research community and as role models for younger women scientists. I would also like to take this opportunity to wish Professor Arnon many more years of good health, happiness, and generosity on behalf of all of you. Now we have completed the formal part of the program of the Adams Seminar. We have learned a great deal from our incredible Adams awardees, and we thank them all for enlightening us. I'd like to conclude again by thanking Sylvan and the Adams family for continuing Marcel's tradition of supporting the best people, supporting our best minds, supporting our future.
And never missing an annual meeting.
And never missing an annual meeting, right. Also, for the next 10 years. Why 10? At least until the age of 93, right? I think that's the last time that your father came. He was 93. Okay, and I would like, of course, to thank also the leadership of the Israel Academy and the members of the steering committee, the Adams Fellowship Steering Committee of the Israel Academy.
It does great work, led by you, and which does exceptional work, and you pick the absolute best, as I said, best and brightest minds that we have in the country.
It's one of the best. One of the best things that we can do as scientists to see how outstanding our young people are and how promising they are, and probably better than our generation.
And as you know, another aspect of my father's design of this program was that you had to study in an Israeli university. So, of course, you could go and do your postdoc somewhere else. But the idea is to encourage education here in Israel. And we hope that if you eventually choose to do a postdoc elsewhere, that you remember to come back home. It's the best place on earth.
That's home. I can only second Sylvan. There's nothing like home. And of course, I would also like to thank Mayor Bandwine, who is the beating heart of the Adams Fellowship Program, for everything that's happening here and mostly not here behind the screens. Some of you already have been communicating with her. Some of you will surely be communicating again. She's here to help you, answer your questions, and make sure that you sail through your PhD in the best possible way, and only do great things to Israeli science.
I can also add, she's the glue of the program. And so she's here to take care of all of you.
Okay, thank you, Maria.